Hey guys, welcome back to One Stop Biology. So today we are going to discuss about transcription. So guys, let me remind you that we are right now at molecular basis of inheritance, which is the second unit of genetics, and we have covered, a lot of important, very important topics from your boards and NEET exam point of view. We have discussed what is the, detailed structure of DNA, what was the experiments that proved that the DNA and RNA are the genetic material and nothing else. And then in the last video, we studied all about replication as per NCERT syllabus. So, guys, again, I would repeat that we are right now running the NCERT series and we are covering all the topics as per NCERT syllabus. So, before we start with transcription, guys, please do not forget to like the video if you understand it, and please do let me know if you have any doubts. Your feedback is very, very important. So do let me know in comment section whether you like the video or you have any doubt. And please do subscribe to the channel if you are new to my channel, right? So let's start with transcription. So guys, basically the way we studied about replication is that, the entire segment of DNA, the entire DNA replicates or copies itself. Transcription is basically the process of copying genetic material from DNA into RNA. So basically, if you remember the central dogma, it was like DNA which keeps on replicating, right, itself. And this DNA converts into RNA and this converts into protein, right? So this process of conversion of DNA into RNA is transcription. This is transcription. Now, guys, remember that the principle of complementarity. So we studied that, the purines and pyrimidines, the nitrogenous bases complement, right? So here as well, the complementarity basically governs, except for the fact that adenosine basically complements with uracil, right? So adenosine in the case of RNA complements with uracil instead of thymine, right? But unlike replication where you have the entire DNA, the entire DNA of the organism duplicated, in transcription, only a segment of DNA, right? And only one of these strands is copied into RNA, right? So this necessitates defining the boundaries that would basically demarcate the region and the strand of DNA which would be transcribed. So it's unlike, how the DNA replication occurs, wherein the total DNA of the organism gets duplicated, right? In transcription, only a segment of DNA and also only one of these strands is copied into RNA, right? Now, basically, why would you ask me why both the strands are not copied? It is because if both strands act as a template, then there would basically they would code for RNA molecule with different sequences, right? And this in turn, if they code for proteins, the sequence of amino acids in the proteins would be different, right? So that is why one segment of DNA would be coding for two different proteins, right? Now, this would complicate the genetic information, which is basically getting transferred, the machinery of, transfer of this genetic information. Now, that is one thing that, the system would be complicated because both these strands would be, you know, coding for different kind of proteins. Now, the other thing is that two RNA molecules, if they are produced simultaneously, and that would be complementary to each other, right? So that would form double-stranded RNA. Now, if we have a double-stranded RNA, then the RNA would not get translated into protein, right? So the entire process of transcription would be of no use, right? Because you cannot make protein out of double-stranded RNA. RNA is always single-stranded, right? So that is the reason, why you have transcription in just one strand, not in both the strands of DNA, right? So now let's move on to the transcriptional unit. Now, a transcriptional unit of DNA is basically defined by three regions of DNA, right? So there are three regions in DNA, through which the transcription unit is defined. The first one is promoter region. First one is promoter region. The second one is structural gene. Structural gene. And the third one is terminator region. The third one is terminator region, right? Now there is a convention in defining the true strands of DNA, right? So basically, since two strands have opposite polarity, so one is basically from 5s to 3, the other one is 3s to 5, right? So what happens is that we have DNA dependent RNA polymerase. Now we have DNA dependent RNA polymerase. Now, this basically catalyzes the polymerization in only one direction, which is 5 to 3, right? So, this enzyme catalyzes the polymerization in only one, one direction. So, basically, the strand that has polarity 3 is to 5. Remember, the strand that has polarity opposite to this one will be the template, right? So that will be the template strand. So the template strand will have polarity from 3 is to 5, right? Now the sequence will be same as RNA. It's just that thymine will be replaced by uracil, right? Now, this basically the sequence which is same as RNA is displaced during transcription. Now, what happens is that the strand which does not code for anything is referred to as coding strand, right? So remember that there is one which is template strand, the other strand which is not coding for anything is basically made up with the coding strand. It is referred to as coding strand. So all the reference point while defining a transcriptional unit is made with the coding strand itself, right? So say, I'll give you an example as well. Now say we have a hypothetical sequence here, right? So this is the sequence, right? Now here if you see, this is the template strand. Why? Because it is running from 3 is to 5, right? And the other strand will be considered as the coding strand. The other strand will be the coding strand, right? Now what happens? Let's see. Now the promoter and terminator. There are two, right? There's one structural gene, and apart from that, there is one promoter and there's a terminator, right? So the promoter and terminator basically flank the structural gene in a transcription unit. So if I show you the diagram as well, if you see this, this is a transcription unit, right? Now in this, basically say this goes from 3 to 5 and this goes from 5 to 3. Now it means that this 3 to 5 is the template. Oh my god. Yeah. Template strand. And the other would be coding strand, right? Now if you see the other thing, so this basically is the promoter region and this is the terminator region. And this entire thing is the structural gene. So this is the structural gene. Now basically this is the site, promoter region is the site where transcription starts. Promoter region is the site where transcription starts. This will be the transcription start site, right? Now what happens is basically so we can say that the promoter is located towards 5 end of the structural gene. Promoter. Now what I said is that whatever we'll be discussing will be the point of reference will be the coding strand, right? So here we can say that the promoter region is located towards the 5 end of the structural gene, right? Upstream. Now, basically, it is a DNA sequence that provides building site for RNA polymerase, right? And the presence of promoter region in a transcription unit also defines what would be the template and what would be the coding strand. If we switch the promoter's, position with that of the terminator, the definition of coding and template strand will be reversed. So if I basically switch these positions, then the entire sequence will be reversed, right? So the coding and the template will be reversed. Now the terminator is located towards 3 end, which is downstream of the coding strand. So again, I'm talking with reference to the coding strand. So the terminator is basically located towards the 3 end of the coding strand, and that defines end of the process of transcription. So this basically defines start of transcription, and this defines end of transcription. Now there are additional regulatory sequences as well, which basically may present further upstream and downstream to the promoter, right? Now let's see what is a transcription unit and the gene basically, right? So basically a gene is defined as Mendel said, functional unit of inheritance, right? Gene is defined as functional unit of inheritance. So basically, there is no ambiguity that genes are located on the DNA. We know that the genes are located on the DNA, right? But it is very, very difficult, to define where it is located in terms of DNA sequence, right? So a DNA sequence, say coding for a transfer RNA or ribosomal RNA molecule also define a gene, right? But by defining a cistron, and what is a cistron? I'll tell you that. So basically, by defining a cistron as a segment of DNA coding for a polypeptide. So basically, say cistron is a segment of DNA coding for polypeptide. The structural gene in a transcription unit could be said as monocistronic, right? Or polycistronic. Now, in case of basically what we can say is that the structural gene can be monocistronic and polycistronic. Now, this happens in the case of eukaryotes, and this happens in the case of bacteria and or prokaryotes. Right? So in eukaryotes, the monocistronic structural genes basically have interrupted coding sequences. The genes in eukaryotes are split, right? Now the coding sequence or expressed sequence are known as exons. Remember, coding sequences are known as exons, right? So exons are said to be those sequences which appear in mature or processed RNA. Now these appear in mature or processed RNA, right? So basically the coding sequences, right, are or the expressed sequences are, we'll call it as exons. Now, exons are basically sequence that appear in mature or processed RNA. Now, these exons are interrupted by introns. So you have introns in between. Now, introns are interfering sequences which do not appear in mature or processed RNA. Right? So the split gene arrangement basically complicates the definition of gene in terms of DNA segment, right? Now, basically the inheritance of a character, if I inherit a character, it gets affected by promoter and regulatory sequence of a gene, right? So now let's see what are the different types of RNA and what is the process of transcription. So we have basically heard of few terminologies which are introns, exons, cistron, right? And monocistronic, polycistronic. Now we know that all eukaryotes are monocistronic, or most of the eukaryotes are monocistronic, and most of the prokaryotes and bacteria are what? They are polycistronic, right? So let's see what are the different kind of RNAs and what is the process of transcription. Now, if we talk about bacteria, in bacteria, basically we have three major type of RNA, three type of RNAs, right? We have the first one as mRNA, which is nothing but messenger RNA. Then the second one we have is tRNA. This is transfer RNA. And the third one that we have is rRNA, which is ribosomal RNA, right? Now, all three RNAs are basically needed if we want to synthesize a protein in the cell. Remember, all three are needed to synthesize protein, right? So the mRNA does what? It provides the template. The messenger RNA provides the, you see, so you will have to refer to the name. What is the term given? Messenger RNA, right? So it provides the template, right? Transfer RNA. This brings amino acids and it reads the genetic code. So it is transferring something. What? It is transferring amino acid and then reading the genetic code. And the third is rRNA, right? So basically this plays role in structural and catalytic. It plays structural and catalytic role where during translation. And guys, translation is the process when RNA is converted to protein. When RNA is converted to protein, the process is known as translation. So the messenger RNA provides or serves as template. It provides the template. The transfer RNA brings the amino acid and reads the genetic code, and the ribosomal RNA basically plays structural and catalytic role when the RNA gets converted to protein. When the RNA, basically protein is synthesized by the RNA, right? So these are the three different types of RNA. Now there is the enzyme which is DNA dependent RNA polymerase, which catalyzes transcription. So we have RNA polymerase. The way we had DNA polymerase, we have RNA polymerase. This is basically DNA dependent. This is DNA dependent RNA polymerase, right? So this catalyzes transcription. This catalyzes transcription of all types, right? In the bacteria, in the RNA, basically of RNA in bacteria. Now, this RNA polymerase binds to the promoter and initiates transcription. This process is known as initiation. So let's do something. right? So first thing one, what we said is that we have RNA polymerase. So say this is the DNA helix. This is the DNA helix, right? Now you have 3 is to 5 strand and you have 5 is to 3 strand, right? Now here, the first part, you have RNA polymerase, right? This is basically known as sigma factor. It is part of RNA polymerase. And here, this region is nothing but the promoter region, right? So the first step is that RNA polymerase binds to this promoter region and initiates transcription. Now, guys, this process is known as initiation. It is basically initiation of transcription. So this process, when the RNA polymerase binds to promoter and initiates transcription, it is initiation. Now it uses nucleoside triphosphate as substrate. It uses nucleoside triphosphate as substrate and polymerizes in a template which is dependent on fashion, which is basically which is basically in a template dependent fashion, and it follows the rule of complementarity, right? So it facilitates opening of the helix and continues elongation. So it opens the helix and then only a short stretch of RNA remains bound to the enzyme. Now once the polymerase reaches the terminator wala region, the nascent RNA falls off, and along with that, the RNA polymerase also falls off. Now, that process is known as termination of complex. So, here what you are seeing, the next step basically is this is the RNA being synthesized. Remember, right? Again, this is the 3 is to 5 wala region, and this is 5 is to 3 wala region. Now, you can see the helix opening, RNA getting produced. So basically, here you can see DNA elongation, right? And then the RNA falls off. And along with that, the RNA polymerase also falls off, right? This is known as rho factor. This is known as rho factor, right? So when it falls off after reaching the terminator region. So guys, again, this will be the terminator region, right? So after reaching the terminator region, RNA will fall off along with RNA polymerase. This process, basically the process getting terminated, is known as termination. So basically there are three steps in transcription: initiation, elongation, and termination, right? Now basically what is happening is that the RNA polymerase is only capable of catalyzing the process of elongation. It basically it basically gets associated with some initiation factors. So this, sigma or rho that you are noticing are what? These are initiation. So one is initiation factor and the other one is termination factor. So see, the what I mean to say is that the function of RNA polymerase is to elongate. Now, along with with these factors, like it uses sigma factor during initiation and it uses rho factor during termination. And along with these factor, after getting associated with these factors, it starts and terminates the transcription process as well, right? So association with these factors basically alters the specificity of RNA polymerase to either initiate or terminate. Now in bacteria, since mRNA does not require any processing to become active, and also since transcription and translation takes place at the same compartment, what happens is basically uh, the translation begins much before the mRNA is fully transcribed. So basically the transcription starts subsequently as transcription is happening, right? But in case of eukaryotes, let's see what happens in case of eukaryotes. Yes. So basically, in case of eukaryotes, there are some complexities. Now, one of the complexities that there are at least three RNA polymerase in the nucleus, right? So basically, in addition to the RNA polymerase which is found in the organelles, there are three additional polymerases. There are at least three polymerases. Now, basically, what happens is this basically shows that there is a clear-cut division of labor. So we have RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. Now RNA polymerase 1 transcribes, here you can see RNA polymerase 1 transcribes rRNAs, ribosomal RNAs. So 28S, 18S, and 5.8S. And RNA polymerase 3 is responsible for transcription of tRNA, smooth, and small nuclear RNAs, right? So it is basically responsible for, the first one is responsible for ribosomal, the second one is responsible for transfer and smooth nuclear RNAs, and then we have two, right? So RNA polymerase 2 transcribes the precursor of mRNA, which is hnRNA, heterogeneous nuclear RNA. So now the function of all the three polymerases are clear. Now this is in case of eukaryotes, guys. So this is one complex behavior. Now the other complexity is that the primary transcripts contain both exons and introns. As we said that they are monocistronic and they have the transcribing wala segment and non-transcribing wala segment, right? So basically the transcript, the primary transcript, they have both exons and introns, and these primary transcripts are not functional. They are non-functional. And that is why what happens is they undergo subsplicing first. And what happens in subsplicing basically, in during splicing, the non-transcriptional segments, the introns are removed and exons are joined in a defined order. The introns are removed and exons are joined in a defined order. Now the heterogeneous nuclear RNA that undergoes additional processing, which is caping and tailing, right? So in caping, what happens is an unusual nucleotide, which is methyl guanosine triphosphate, is added to fifth end. And in tailing, what happens? In tailing, adenylate residues, right? So 200 to 300 adenylate residues are added at three end, and that is added in a template independent manner. Now this becomes the fully processed heterogeneous nuclear RNA, which is now known as messenger RNA. Now this is transported out of nucleus for translation. So there is again another complexity in case of eukaryotes, right? Now the significance of such complexities, we'll understand it in the later videos, but now you can see that the presence of introns gives one complexity, that you have to splice it and you have to join the exons in a proper manner, right? So basically all these things makes the transcription process very very complex in case of eukaryotes. So guys, with this we have finished the topic of transcription. Again, it is very very important, but as I said, it is very complex, and sometimes one feels it very difficult to understand this. So please do let me know if you did not understand any part of this video, or if you want more clarity or on any subtopic of transcription. And please do like the video if you understood the topic. Share it with your friends, and please do not forget to subscribe to the channel if you are new, for all the updates and all the details, important details that I'll be posting on this channel. Thanks guys. See you in the next video. Bye bye.